Let me share my screen. We don't need to see that. I don't think we need that. Well, this is something I can show you. Hopefully you've taken a look at our website. If you haven't, I think you have to because you had to log in on Zoom on the website. But take a look at it. I've got a number of things. Please read, note. There's the syllabus. Something about student and technical help. Some videos for class. This isn't terribly important. I'll have a link to all the videos we're using within each lesson, but if you want to look at the videos of some of them for class. The email messages I sent you. Here's an extra credit study sheet. Let me open that up. It's worth 6 points, and it's due June 12th, so you've got about a week to get that done. What you do is, where the heck is it? I don't have a link for it. Well, I'm glad I checked. Let me see if I can find that quickly. If I don't find it quickly, we won't do this. Remove that. And move that over. Okay, it's just going to show me all my documents, so that's not going to be quick. All right, I'll try and get that link on here to where you can correct that. I made a note of to myself. I thought I checked all this last night doing all of that, and obviously I missed this one. Make link to extra credit study sheet. What you're going to be doing is just turning in a worksheet, Microsoft, what do you call that? Microsoft Word, Excel spreadsheet, detailing when you're going to be studying. If you're going to be studying all the same, you're planning on studying all the same all term long, you only need to fill out the second tab of the worksheet. The first tab of the worksheet is just for you to figure out when you're working, when you're going to be eating, when you're going to be sleeping. So it's the general things. And then on the second tab, all you do is you put on your when you're going to plan to be studying for this class, the lecture and the lab. And if you need to put different weeks that you'll be studying, because I don't know, maybe you're going on vacation during the second week, you would put that on another tab. And you can just keep adding tabs if you need to. If you're all planning to study the same, then you just put it on the second tab. And that'll be a little more clear once you've done it. And then submit it to this link or this folder in Canvas here. And that'll be extra credit. Let's go on. There's another extra credit assignment, since I'm talking about extra credit assignments. Let's go into week 1. There it is. Send me your picture. It'll go in my gradebook. I want to learn your names. And for me, the easiest way to learn your names is to see your name when I'm recording the grades. And so nobody will ever see this except for me. Make sure to picture your face. If you don't have a picture that you can send me, like a digital copy, just get somebody with a phone to take a picture and then email it to me, either the Canvas website or my course website. And I'll get you extra credit. I think that's for 3 points. Yes, 3 points. And mail it to the folder in this this folder in Canvas, sorry. So you're not really mailing it, you're sending it to the folder in Canvas. They can email it to you and then you can put it in the folder. All right, what else do we need to talk about? The quizzes, as well as the final, will be requiring you to download something called Lockdown Browser. And I think it has Respondus Lockdown Browser. So to take the quizzes, and as well as to take the final, you need to download this. I suggest you do it before a quiz, and I've got, what do you call those, some extra credit quizzes. They're going to be practice quizzes, and I'm giving you extra credit for the quiz. I suggest you download it at least by that time and get it running on your system. Every once in a while, about every other term, I have a student, so it's not usually more than 1 or 2 students every other term, who have trouble getting it to run on their system. And if you have trouble, contact the computer help desk. I don't remember what it's called at Yavapai, and they are used to getting this up and running on your system, so they can help you getting it running. But you don't want to wait to download it right before you have to take the first quiz, and then you find it doesn't work on your system, because it may take you a day to get it up and running on your system, meaning you're going to contact the computer help desk and then they'll get back to you, and so it might take a day to get it up and running. Most students don't have a problem getting it running on their system, but like I said, every other term I have 1 or 2 students who have trouble getting it to run on their system. And then a note about running Lockdown Browser to take the quiz. You want to shut down generally everything on your computer except for the Lockdown Browser, so you can take the quiz, meaning it's sort of a, what do you call that, a memory hog? Something like that. And if you have a whole bunch of things open on your system, especially if you have a slow system, it'll slow down your computer, and then when you're taking the quiz, it'll be slow, and you don't want that because the quizzes are timed, and the point is, shut down everything you need, and then it won't slow down your computer, and then you won't have trouble taking the quiz. What you just do is, like this one, I don't need it, so I can shut that down. I don't really need that, so I can shut it down, but I'm not going to because I want to come back to that. So do that when you're taking the Lockdown Browser. This will lock you into this browser while you're taking the quiz, and you'll be locked in until you submit it. And that just helps prevent cheating, meaning you can't open up another browser and then look up the answer. The quizzes are timed. You can look up answers in your notes and in the textbook, but they are timed, so you won't be able to look up more than about 1 or 2 questions. So don't plan on doing that, because you'll run out of time, and then if you don't finish the quiz, I don't know why that ran there. You won't be able to finish the quiz, and then you'll miss those questions. All right, any question about any of that? Let's see. You'll have to take in the first week, so I think it's by July 11th, the plagiarism tutorial and the plagiarism quiz. The quiz is worth 20 points. You can take this quiz multiple times. I think it's something like 5 times. You do need to make 100% on this quiz, or you're going to get 0 on one of the major assignments. The research project, which is the disease research project, which is 100 points. If you don't get 100% on the plagiarism quiz, you'll get 0 on this infectious disease research project. So you want to take it multiple times if you don't get 100%. And it'll be the same question, so you just have to remember what your answer is. Although when you take the quiz, I think it shuffles the question answers, so you can't just always take A, because it'll switch whatever the correct answer is. And there's a guide on writing up APA. I forget what APA stands for. American Physicist or Psychologist citation guide. And then we're not going to be covering Chapter 2 because you've taken that in the previous course, but if you want a refresher, here are the links to Chapter 2. Chapter 2 is the chemistry of life, and I've got my overview and objectives, the study guide, as well as the lesson notes. And then Lesson 1, the overview and objectives, the study guide. This is the material we're covering, but in sentence format. And then later, once we've finished the lesson, I will make the lesson, meaning the slides, available to you. There will be a worksheet for Chapter 1. It's worth 2 points, and it's due. The worksheets are always due on Saturday of the week that we're covering the lesson. And let me mention that the quizzes are always due on Friday of that week when the quizzes are due. Any question about that? I already talked about you can send me a picture, it'll be extra credit. And then later in the week we'll do Chapter 3. We're not going to do that today. Any questions about what we're doing or the Canvas website? All right. I think I'll leave that open and sorry, I'm having trouble. There we go. Let's talk about the syllabus. So, syllabus for Biology 205 for both the lecture and the lab. I should note that I will be here from 1 to... Today I think I'm going to quit early, and that's because I want to make sure that the Panoply recording gets finished before I start the lab at 2:30, and I'm not sure that 15 minutes will be enough time, so I'll try and stop at 2:10 today to give it 20 minutes. And then I'll be back at 2:30 to start you in the lab. And I will be there while you're working in the lab in case you have any questions. We'll be meeting for the lecture on Monday and Wednesday and Tuesday and Thursday. And, I don't think we're meeting till 3:45 in the lecture. I'll take a look at that. But we're meeting at the normal lecture times on Monday and Wednesday and Tuesday and Thursday. We'll have the lab twice a week from 2:30 to 3:45 on Monday and Wednesday. All the recordings will be recorded, so if you miss it, you should be able to view it from the Canvas website, and hopefully, let me show you that. You guys are probably familiar with it from seeing it in another class. I'm not familiar with it. I've never used it before. So if you come on over here, and I've got more links than you'll have, but the Panopto recordings, you should be able to click this link and get there. There will be nothing there yet, and I'm not sure how soon it will be there, meaning this is all new to me. I've never used Panopto before, but they told me that until I get the first class recorded, they won't be sending anything to the Panopto recordings. Because I asked about that. I did a test run and there was nothing there. And all right, I will also have them on my where do I have that? Maybe the video class link. I'll have them on my YouTube site, at least initially, because I'm not familiar with Panopto. Later on, I may discontinue putting it up on the YouTube site, meaning if I like the way Panopto is doing it, I'll just put them only here. But for the time being, I'll put them at YouTube because that's what I'm used to doing. All right, the textbook material is the Microbiology and Introduction by Tutoria, Funk and Case. And can somebody tell me, if you got the textbook, was the custom version of the textbook available at the bookstore? Can somebody tell me that? So everybody who's got the textbook, you got the regular full edition of the textbook, is that the case? All right, so it looks like the custom edition of the textbook is not available. One good thing about the full edition of the textbook is that we will be doing a few chapters which are beyond the custom edition of the textbook. So I've got to work on that. Meaning I don't even know if antibiotics, host defenses 1 and 2 are in the full edition of the textbook. I've got to look that up. But I know parasites are, and then classification are. And so those will be in the full edition of the textbook. I'm going to guess antibiotics are also in the full edition of the textbook. So you'll be able to look up something if you have questions. I have no reading for all of this section because it's not in the custom edition of the textbook. But we are doing these last chapters, and I decided to have the custom edition of the textbook because the custom edition is considerably cheaper than the full edition of the textbook. But if you can get the textbook used online, it's pretty reasonable. And it's not that unreasonable if you get the used edition of the textbook from the bookstore. All right, let me go back to the syllabus. I was hoping the custom edition would be available because I put that in, gosh, I would say about 3 months ago. All right, there are some optional tests. You can read about that. You should know about the other resources that are available, mostly on my Canvas website. But Yavapai has a college website. You should be familiar with that. And you should learn about your responsibility to visit the Canvas website. If something is changed, I will email you as well as put an announcement on Canvas. So take a look for those. I'm not going to discuss the course description. You can read about that, the course policies and suggestions. Oh, actually, I will talk a little bit about the course policies and suggestions. If you're sick, contact me and I'll help you keep track for the class. And if you're sick and you're not able to make an assignment by the due date, contact me and I will change your due date. Okay? But you have to contact me before the due date to do that. Preparation for class, you should be getting prepared for class, like reading the material beforehand. And this class is going to be very busy. So you must pre-read the material, like the lab before the lab, and the textbook before the quizzes. You should also review the material before the quizzes and the labs. Attendance will not be taken in class. Maybe later on I'll start taking attendance and give you extra credit, but it's actually difficult to do. I've got to go through each student and then record them, and so we'll see about that. You must take the online quizzes by the due date. The online quizzes will be open at 11:59 p.m. Thursday night, and you must take the quiz by 11:59 p.m. on Friday. If you're in the middle of the quiz and it becomes 11:59 p.m., the quiz will automatically time out and you'll be done with the quiz. So make sure you're finished by 11:59 p.m. on Friday. The purpose for having the quizzes in 24 hours is to reduce the cheating. It's kind of difficult to cheat because each question will have a selection of questions, and so you'll be answering generally different questions from other students, but some of them will be the same, and I don't want students to tell each other what's on the quiz and what they selected for an answer, and so you have to take your quiz within 24 hours. Any question about that? And I selected Friday because that's not a day that we're meeting, so you'll be generally a little more free on Friday, and you can take the quiz anytime during that 24 hours. But it must be taken within that 24 hours. Any question about that? All right, so you should read about the student responsibilities, know about course withdrawing. That must be done with the students, except I think in the first week I can actually withdraw students or drop them in the first week. Late assignments, I do encourage you to turn in all assignments even if they are late. However, there is a penalty if you turn it in late. If you turn it in 1 day late, the penalty will be 25% for the first day. After that point, the penalty is 50%. So turn in your assignment by the due date to avoid getting the late penalty. You can read about student conduct. I'm just going to say that plagiarism is not allowed in Yavapai or school. If you plagiarize, that's use somebody else's work, usually their words, you gotta put it in quote and then say who you got it from. Otherwise, it is plagiarism. If I find plagiarism, you'll get a 0 on that assignment. Usually what happens with plagiarism is 2 students will be working together and then they submit the same work. That is plagiarism. So make sure all your work is in your own words, or put it in quotes and say who you got it from. I encourage you to study together, but if you use somebody else's answers, put it in quotes and say who you got it from. Okay? Otherwise, you'll get a 0 on that assignment. You can read about etiquette in Zoom. I've got it set up so immediately when you log into Zoom, you'll be muted, and to say something, you need to unmute yourself. And then when you're done talking, go ahead and mute yourself, because that will keep noises, background noises, from coming over the class. And I should ask, how is this setup doing? Can everybody hear me well? I've got a special, good. I got a special, what do you call that? Whatever this headgear is. For the suggested study methods for this class, you're going to be meeting for about 7 hours a week. You're also going to be meeting or studying, doing your reading, doing your assignments, reading through the lab. And together, all of this will take about 14 hours of time outside of the class time. So the total class is going to take about 21 hours of time. If you do not have the time to spend on the class, and this will be for an average student, well, maybe you should consider taking the class during a semester. That should be a semester, not a quarter, when you have the time to spend on it. Some people who are really good can spend less time on the class, but generally you will be needing to study, to do the reading, and then to do the work, and it'll take you on about, average student, about 14 hours plus the 7 hours we're spending in the class. And yes, that's a lot of time, but this is an advanced class, and this is expected of you. If you don't have that time, like I said, really you should consider thinking about taking the class when you have the time. There's some other information you can read about, like to find out how you learn. You can take this questionnaire. There's some help up here for suggestions for studying. You can read through that. I don't want to go over it in the class. Let me talk about the grading. I've already mentioned the plagiarism quiz is worth 20 points. You can take it at least 5 times. You do need to get 100% on this. Otherwise, you'll get 0 on the, where is it? The Infectious Disease project, which is worth 100 points. There will be 5 quizzes throughout the term. Each one is worth 75 points. The lowest quiz will be dropped, so only 4 of them will count for 300 points. And you'll notice on my website, I think it's quiz 3, it's not worth 75 points. That's because the total of the quizzes is going to be worth only 300 points. So that quiz will be worth 75 points. It's just that I've got to manually do that. And then there will be a column where I'll be subtracting your lowest quiz. And I think that's called Q correction or something like that. Q core. There will be worksheets in both the lecture and the lab. There will be about 1 worksheet in the lab for each lab for a total of 50 points. And that makes about each worksheet in the lab about 5 points. There is an exception. The first 2 for lab 00 and lab 1, they're going to be worth 2.5 points, but all the others are worth 5 points. There will be a lecture worksheet for each lecture. A few of the lectures have 2 worksheets per lecture. They will be worth a total of 30 points, or about 2 points for each worksheet. All of the worksheets are due Saturday when we're doing that material. Okay, so the Saturday of that week that we're covering that lesson. There will be an identification of an unknown bacteria in the lab, and we'll talk more about that when we get closer to it. but that lab project will be worth 75 points. It will be all done online. It'll be a virtual lab project. You'll also be doing an infectious disease research project. This is an 100-point lab project. And you will research an infectious disease and do research on it. More information will be contained in a separate package. We are a really big class, so I'm not sure how I will do this. Normally I require all students to do a different infectious disease research project. Because we're such a big class, I may allow 2 people to do the same disease as long as they cover a little different aspect of the disease. Okay? And I'll think about that before making a decision. The cumulative final exam is worth 150 points. It'll be mostly multiple choice. There will be a few fill in the blank questions or essay questions. 20% will be from the lab and the identification of the unknown. 50% from the material from the last quizzes, so the last 2 weeks of the class. Let me scroll down to the schedule here. So our last quiz will be covering Chapter 7, Antibiotics, and Lab 7. So all the material since that material will be primarily on the comprehensive final. And so that'll be about 2 weeks worth of both the lecture and the lab. That's going to include Lab 9 and Lab 8 will only be on the final, as well as everything since that last quiz. And that's what I mean. Any question about any of that? Where were we? But 30% of the material will be from the earlier material in both the lecture and the lab. Okay, participation really doesn't apply. Here's a breakdown of the points. 20 points for the plagiarism quiz, 300 points for the quizzes. You get to drop 1 quiz. Lab assignments, 50 points. Identification of the unknown, 75 points. The infectious disease project worth 100 points. And 30 points for the lecture worksheets. The final will be worth 150 points. And hopefully that added up to 725 points. I'll have to recheck that. There will be some extra credit assignment points. That'll be about 30 points of extra credit. I pointed out 2 of them in the Canvas website. There will be a 3rd coming up during week 2, and I'll point that out when we get to week 2. There will also be a few extra credit questions on the quizzes, so total together about 30 points. The grading scale, if you get 90% or better, it's an A, 80% or better, it's a B, etc. Down to a D, which is 65% or better. I'll let you read about the question review. Proper microscope use, you're not really going to be working with microscopes. You can read about the non-discrimination policy. Safety information since students and physicians, you can read about that. It's not terribly important because you won't be working with real live microbes. Words of advice and comfort, I'll let you read about that. Lab safety rules, you don't need to follow that because we're not really going in the lab, but you do need to know about lab and safety rules in microbiology, so you will be tested on this material, So you do need to read about it and understand it. If you have any questions, please ask me. I'm not going to go over it here, but there's a bunch of rules on working in the lab. We'll go over that when we're talking about the lab. And then at the very bottom of the syllabus is the summer schedule. It is tentative, meaning I might make changes. I don't think I will, but I might. And if something happens, I'll make a change and I'll let you know. But on the left, we have the lecture topics and quizzes, and on the right, the laboratory topics. There are no quizzes in the lab. They'll be done in the lecture. So today we're going to cover, obviously, the overview of the syllabus. We're going to start Chapter 1, the microbial world and you. That does include a part of Chapter 10, the classification of microorganisms. We'll go into a little bit more near the end of the class about classification also. Let's see, that's it for the first 2 days of class, and then I'll talk about the lab in the lab. But today we're going to be doing Lab 00, the lab safety training. All right, any questions about what we're doing and the schedule? The quizzes will be highlighted in blue, the practice quizzes in yellow. The practice quizzes will be for extra credit points. Anyways, it's got the assignments when they're due. The worksheets are not in the schedule, but every worksheet is due at 11:59 p.m. on Saturday of the week when we're doing the material for that worksheet. All right, any questions about what we're doing? Any questions about the syllabus?
I have a question about the worksheets.
Uh-huh.
In Canvas, it says something about it being a class discussion, and I just wanted to know how we're supposed to upload our worksheets.
You go to the link. I won't quite be able to show you because I'm an instructor. I don't have that link. And on the right or on the bottom, actually for the lecture worksheets, you click this link and then it gives you the worksheet. But for the lab worksheets, when you click this, either on the right or on the bottom here, there will be a link for getting the worksheet. Okay? And then on your site, on the right here, if you have the room, or on the bottom if you don't, there will be a link for submitting it. I don't have those links because I'm an instructor. Okay? All right, hopefully that answered your question. Any other questions? If not, let's move on to Chapter 1, the microbiological world and you. And Chapter 10, if you have the current version of the textbook, I believe it's from page 272 to page 281, but it's covering the classification of microorganisms, if that isn't the correct page in your textbook. Generally, I give a slide giving you the major goals and a rough outline of what we're going to be covering. So for every lesson, I try and do that. And here it is here. The major goals, well, know the major terms of the chapter. And this is the major terms here, but it's not all of the terms. So if you want to find all of the objectives, let me see if I can get that. For all of the objectives, you need to click on the link for the lesson, and that would be the overview and objectives for Chapter 1 doc. On some of them, I've got the study guide and the objectives together. So let's go down later. There we go. So this would have the objectives on the top of the study guide. But that's where you find all of the objectives. Any question about any of that? So this is only the main goals, the major objectives. And then the rough outline, know the terms, understand a brief history of microbiology, and understand the importance of microbes to you as well as the Earth's ecosystem. Any question about any of that? Okay. So let's begin talking about the importance of microbes. Here we are looking at the nitrogen cycle. And if you don't know, nitrogen moves around in the Earth's ecosystem. So nitrogen can start in the atmosphere, and it can be fixed by bacteria and archaea into organic compounds in the bacteria and archaea. And then when they die, decompose, bacteria and archaea and fungi can decompose the organic compounds and change it into ammonia. It is true some of the organic compounds can be taken up by the soil from something that's dead, and the plants can use those directly, and some can be converted into nitrite, but most of it is converted into ammonia. Ammonia and ammonium, that will be a conversion of ammonia just by water mixing with ammonia, can be taken up by the plants, but plants don't like ammonia. They can take it up, but they really don't like it, but they can use ammonia or ammonium for a nitrogen source. But the ammonia or the ammonium can be nitrified by bacteria and archaea into nitrite, and then the nitrites can be changed by bacteria into nitrates, and the nitrates can be taken up by the plants in the soil, and plants love nitrates. If you look at plant fertilizer, it'll have nitrates in there. Plants love that source of nitrogen, and they can use it and then take that nitrogen, either the ammonia or the nitrates, and make organic compounds out of it. And most of the organic compounds, when the plant dies, it'll decompose and it'll be broken down into ammonia or ammonium. But like I said, some of it can be an amino group, which the plants can use directly, and some of it can decompose into nitrates. The important part is that nitrogen can move around, and from nitrate can be converted by bacteria and archaea back into nitrogen in the atmosphere. And so that's how nitrogen can move around in the nitrogen cycle on the planet Earth. How important are microbes in our lives and with the planet? Well, let's look at the nitrogen cycle. Wherever you see a red arrow, this step in the nitrogen cycle is happening because of a microorganism. So the reason why this step in the nitrogen cycle is happening, like for example, converting nitrogen into organic compounds, that's because of a microbe. And as you notice, almost all of the steps of the nitrogen cycle are happening because of microbes. Like there's this step here, that one's not happening because of a microbe, or that step there. But most of them are happening because of a microbe. So that microorganisms are extremely important for the recycling of nitrogen in the planet Earth. We would not have the nitrogen cycle happening if the microorganisms weren't around and to run the nitrogen cycle. Any question about any of that? All right, so microorganisms are important. Microorganisms are also important in biogeochemistry. There are rock-eating microbes, where some microbes actually obtain their nutrients directly from rock. They break down the rock, leading to the formation of soils. And all of these pictures are trying to show you pictures of where microbes can break down the soil. Now, microorganisms are not the primary way that soil is made. Soil is primarily made by erosion from wind and water. But microbes can make a significant portion of the soil, so they are important for the formation of soil. If we look at the Earth's oxygen levels over time, you'll note that the Earth oxygen level changed. Initially, there was no oxygen in the atmosphere on the planet. And then about 2.7 billion years ago, oxygen started accumulating in the atmosphere. And then there was a slight increase up until about here when oxygen levels really increased until we have today where the oxygen level is about 21%. Okay, how did the first oxygen come about in the planet Earth? Well, it was microbes. The cyanobacteria evolved, and in their photosynthesis, they created oxygen, actually as a waste product, and that oxygen came out and started accumulating in the atmosphere. And because of them, it went to about 5%. That allowed green plants to evolve, which happened about this point. And today, we have about 50% of the oxygen in the Earth's atmosphere, how do you say that? Responsible, it came about from microorganisms. The other 50% of the oxygen is from green plants and algae. So only about half is from green plants. The rest of the oxygen, about 50% of that oxygen, is due to microorganisms. Once again, telling us how important microbes are to the planet. First of all, without the microbes, we wouldn't have had the evolution of plants and animals. And then if you don't know, we require 21% oxygen to survive. So we need the microbes to make us oxygen. Any question about any of that? All right. Let's go on and talk about sewage treatment plants. Humans are responsible for making the waste, right? And here we can see the human sewage treatment plant, and it was obviously made by humans. So humans are responsible for breaking it down. Is that right? It's a yes or no question. Are humans breaking down the waste in this waste treatment plant? Yes or no? You
No.
got a 50% chance.
Yes.
Yeah,
No.
the correct answer is no. The microorganisms break down the human waste. We generate the waste when we flush our toilets and wash something down our sink. It accumulates here. But then in these treatment plants, microorganisms break down that waste material generally into carbon dioxide and water and recycle the waste material. So the microorganisms are important for breaking down these wastes. So there are a number of ways where we see that microorganisms are extremely important on the planet for running such cycles as the nitrogen cycle, for putting oxygen in the Earth's atmosphere, for making soils, and then for recycling waste material. All right? Any question about any of that? All right, let's talk a little bit more about microorganisms. When we use the term microbe, it will be used synonymously with the term microorganism. Now, what do we mean when we talk about a microbe? That's an organism or a virus that is too small to be seen with an unaided eye. These are the single-celled microorganisms that can take on many shapes and sizes. They include the bacteria, the archaea, the yeast, the molds, the protozoans. Some of the algae, if they're single-celled algae, that will be a microorganism. Obviously, the large algae we see on the ocean beaches, that would be kelp, and that wouldn't be a microorganism. Viruses and prions are also considered microbes, but please understand that they are special because they are not cells. They do not have cells, and you could argue they're not an organism because they don't have cells. Any question about any of that? Okay. Microorganisms are almost everywhere, so we can talk about them as the ubiquity of microorganisms. You will find them almost everywhere on the planet. They play beneficial roles in numerous processes. We've talked a bit about that. And they are numerically the most abundant life form on the planet Earth. What is a cell? We talked about cells. A cell is the basic structural and functional unit of all organisms. It's highly organized. It's a compartment bounded by a thin, flexible structure called the plasma membrane or the cell membrane. All cells contain three components. Obviously, they have the plasma membrane, they have cytoplasm, and they have DNA. Here we're looking at bacterial cells. All right, here we have a whole bunch of pictures of different places on the planet Earth, different people and animals. Where in these pictures would you find microorganisms in every centimeter? Like every centimeter on the cat, would you find microorganisms? Where in these pictures would you find microorganisms? And if you don't want to answer that one, you can answer, where in the picture would you not find microorganisms? In every centimeter.
I would say everywhere except the volcano.
Everywhere except what?
The volcano, I think.
Okay, that's correct. You'd find microorganisms. Oh, I forgot to mention that. Generally, people, I do not use the, somebody sent me a message. I do not use that because I cannot see it when I'm, all of them. Okay, somebody gave an answer. Not all of them. There is one part that they don't see. I don't use the text messages because I cannot see it when I'm sharing my screen. And also, Zoom is really complex. It has a bunch of things in it, like text messages and emoticons and symbols, and you can go thumbs up or thumbs down and all that sort of thing. And it's really too much for one person to handle. They suggested when I was first learning about Zoom that if you're using all of those features, you have two instructors teaching the class. One person doing the text messages and the emoticons and the other symbols, and then one person lecturing. Well, guess what? You've only got one instructor. And so I'm only going to be using the lecturing, and if you want to send me a message, you have to do it vocally, because I do not see the text messages when I'm sharing my screen. Okay? Any question about that? I think I also don't see the thumbs up and the thumbs down, but I don't remember, meaning when you send me an icon. I've got to get out of the share screen to see that. And generally, I'll be sharing my screen. But everywhere in this picture, you would see microorganisms. Everywhere in this picture, you'd also see microorganisms. And guess what? That includes the air. We have microbes up in the air, about a mile up in the air. Okay? And here, everywhere in this picture, including down into the soil, we have microbes down into the earth, about a mile into the soil. Okay? That will differ if you hit solid rock, in which case I'm not sure how far down it goes then. But with soil, microbes can be found a mile down into the earth. And that's true a mile underneath the ocean floor as well. We find microbes everywhere here, meaning just because it's a desert, there's fewer microbes, but they still are every centimeter. And then everywhere here, except for right here where the lava is. That's too hot for any microbe to survive on. Any questions about any of that? All right, I'm not hearing any questions, so let's go ahead and move on. I'm thinking about if I want to stop here, but I think I'll go on for 2 more minutes. I mean, 10 more minutes. All right, there we go. So we see everything except where the lava is. Microorganisms are almost everywhere. That includes environments on the land and the water, in the atmosphere, subsurface below the land and the water, on plants and animals, essentially everywhere where liquid water is present and temperatures are between a minus 20 degrees Celsius and a minus 120 degrees. Any question about any of that? All right, we do find microorganisms below that temperature, but they'll be frozen. They won't be metabolically active. But above that temperature, that will kill the microorganisms. Generally, the endospores might survive, but most microbes will die at that temperature. So microbes are numerically the most abundant life form on the planet Earth. How many microorganisms are there? In 1 gram of soil, there's about 1 times 10 to the 8th microorganisms, depending on how wet the soil is and how much, what's the word I'm looking for? How much organic matter is in the soil. But generally about 1 times 10 to the 8th, and that's something like 100 million per gram of soil. For 1 millimeter of ocean water, are you going to find more or less microorganisms in 1 milliliter of ocean water? And 1 milliliter of ocean water is 1 gram. Okay, I'm not hearing an answer. Somebody's got to guess. More or less?
More.
More.
Both people said more. Actually, in ocean water, oceans are considered the desert of the planet, it's less. It's one place on the planet where we have less than in the soil. For 1 gram of human feces, there's going to be more. On one human being, there's going to be a lot more. On the entire planet Earth, there's going to be a lot more. And we got the numbers here. So in 1 milliliter of ocean water, there's about 1 times 10 to the 6 microorganisms. And then, depending on the soil, which soil, we have about 1 times 10 to the 8th. In human feces, if it's a fresh feces, we will have 1 times 10 to the 11th. However, if the feces is in the desert and it's dried out, you'll actually only have 1 times 10 to the 6th. So it does depend on how fresh the feces are. And curiously, human feces, about 55% of the wet weight of the feces is from microbes. So that's over half. On a human, you have 3.8 times 10 to the 13th microbes either in or on the human being. On the planet Earth, we have 1 times 10 to the 30th zeros, and if you know that number, it's a nonillion microbes on the planet Earth, or 1 times 10 to the 30th microbes on the planet Earth. If you don't know, the population of the human population is between 7 to 8 billion people. Okay? All right, any questions about any of that? The important part is there's just astounding numbers of microbes, and we're really only counting the prokaryotic cells. If you were to go for the viruses in 1 gram of soil, it would be 100 times higher than this number. And that's because viruses are very common too. All right, microorganisms and disease. Let's talk a little bit about that. There are pathogenic microbes. Pathogenic means the microorganism causes human disease. So a pathogenic organism is a microbe that causes human disease. You should know that most microorganisms do not cause disease. They do not cause human disease, and they don't cause any other disease. Only a small percentage of all microorganisms are pathogenic or disease-causing.
Can you give an example of one of those?
Yeah. Trying to remember the name. Bacillus anthracis causes anthrax. That would be a pathogenic microbe. And then all of the diseases of man would be caused by a pathogenic microbe, like the flu is caused by the influenza virus. Okay? All right, let's talk a little bit more about diseases. We can break diseases into 2 categories. Infectious diseases, which we will be studying in this class, and inherited diseases, which we will not be studying in this class. Infectious diseases are caused by microbes. And we've already given an example of that. Bacillus anthracis causes anthrax, and the influenza virus causes the flu. So all infectious diseases are caused by microbes, and that is why they are infectious, because the microbe can move from one patient to another. Inherited diseases are not infectious, and generally the person is born with them. So depending on what genes they were born with, they can get an inherited disease. Can somebody give me an example of an inherited disease?
Cystic fibrosis.
Yeah, cystic
Huntington.
fibrosis. Another would be sickle cell anemia. These are diseases that the person is born with. And they're inherited, not infectious, meaning nobody else can get that disease from them. All right, let me talk the last slide about microorganisms. They are small. Generally, when we're talking about prokaryote, most of them are about 1 micrometer, and another term for 1 micrometer is 1 micron. That's about their size. Some of them can be a little larger, like E. coli can be about 2 or 3 microns long, but it's only 1 micron wide. They do depend on different shapes and sizes. Let me blow this up so you can see that a little better. Come on. Oh, okay. There we go. So you can see the graph here for how big they are, and then the different sizes. This one here, each cell is actually one of those dots. So this is a colony of microbes, and this parent colony is about to give rise to baby colonies. 1, 2, 3, 4, 5, 6 baby colonies are inside the parent colony. And then that looks like a lymphocyte. The point is that they are small. You need a microscope to see them. They come in various shapes and sizes. Any question about any of that? If there's no questions, I'm going to end here because I want to end a little early to make sure that the video is totally made before I start the lab. And I will see you at 2:30 if you're coming to the lab.
I'm sorry, I missed the beginning of class. Do we have class Monday through Thursday from 1 to 3:30?
Yes. No,
Okay.
it's 1 to 2:20.
And then we have lab from 2:20 to 3:30?
It's from 2:30 to, what the heck is the lab time? Whenever the lab ends.
Okay, I just wanted to clarify. I'm sorry. Thank you.
Okay. All right.